Good morning, Professor Penaki sir.
Yeah, good morning, Dr. Judith.
Yeah, so
Good morning.
welcome, and
Yes.
we will start in another 5 minutes.
Yeah, sure. So let me just confirm that whether you are able to see my screen.
Okay, okay.
Should I share it now and can you confirm?
Yes, sir. You can share it.
Are you able to see this? Hello. Hello.
Yes, sir.
Can you please confirm that whether my screen is visible?
So we can see your screen, but it's not on full screen mode.
Okay.
Now
Uh,
it is.
it is moving.
Yes, now it's moving.
Okay, good. Okay, so I am there. The screen will remain shared, and just let me know when you want to start it. Okay.
Yes, sir. Sure.
Yeah. Thank
Thank
you.
you.
Trupti, Judith,
Yes, sir.
send participant message to immediately join.
Okay, sir. A very good morning to one and all present here. And once again, a warm welcome to the second day of the national webinar on technological advances in applied microbiology. So we will begin today the first session with a lecture by Professor Pinaki Sar. Sir, we welcome you, and I will just give a brief introduction uh, to familiarize the audience here with your achievements. Professor Pinaki Sar is a professor at the Indian Institute of Technology, Kharagpur, India. His research interests include studies on microbial diversity and function in natural and impacted terrestrial and aquatic environments, cultivation dependent and independent approaches to understand the biogeochemical role of natural microbiota, and the development of bioinformatic pipelines for genomic and metagenomic sequence analysis, among others. Professor Sar has a PhD in botany from Banaras Hindu University. He has been the recipient of numerous awards and fellowships, right from his doctoral research fellowship from the DBT Government of India during the period 1994 to 99, and all through the years he has been the recipient of a number of awards. I will mention the latest two. uh, he was awarded the Open Competitive Seed Challenge grant from IIT Kharagpur for setting up the genome sequencing facility in 2016, and the Fellow of West Bengal Academy of Science and Technology in 2020. Sir has a huge body of work published in national and international journals. I have with me a list of around 35 publications from the past 5 years only, and these are in very highly reputed peer reviewed journals, including journals with an impact factor of 7 to 9. Um, Sir has around 2,680 citations and an H index of 28. Professor Sar has undertaken a total of 21 projects as PI and 3 as co-PI, as well as 1 as mentor. I will name a few of the recent extramural projects. Understanding landfill microbiome and application for bioremediation of environmental pollutants, funded by Tata Consultancy Services. This is ongoing from 2019 to 2023. And development and demonstration of integrated instrumentation system for quantitative detection of water contaminants with information database for surveillance of waterborne disease, funded by the Department of Science and Technology, Government of India, also ongoing from 2018 to 21. Um, Sir has supervised 11 doctoral theses completed and 5 continuing, as well as 1 MS thesis. Sir, it is our privilege to have you with us today, and we are looking forward to learning about the deep life underneath the Earth's crust through genomics. Sir, over to you.
Yeah, thank you, Dr. Judith, and a very good morning to everyone. I am really happy to be here amongst you, and I am really happy also to present our work on deep life within the Earth crust. The topic, I don't know, maybe somewhat new to many of you, but I will try to explain the topic as well as the findings obtained during our research in past couple of years. So just to confirm again that you are able to see my screen and hear me clearly.
Yes, sir. We can see your screen and we can hear you as well.
Yeah, thank you. So can I start now?
Yes, sir. Please go ahead.
Okay, thank you everyone. So the topic of the today's lecture is exploring the deep life underneath the earth crust through genomics. And the following layout will be followed with a brief introduction about deep life. I would explain some of the genomic approaches which are considered to be cutting edge and most appropriate to investigate the microbial systems because many microbes they do not grow under laboratory conditions. And then I will take you through our findings on deep life underneath the Deccan traps. Now, Earth is a microbial planet. Why? Because microbes run the world. Now, every process in the biosphere, whether we see or whether we do not see them, whether we understand those processes or we are yet to understand those processes, they seems to be all touched by the seamlessly endless capacity of microbes to transform the world around them. Now, the deep dark biosphere. So as you can understand from the title itself, it's deep, that means under the earth, and it's dark because sunlight cannot penetrate there. So unlike the surface exposed ecosystems, which are often well explored and well investigated for different purposes, interestingly, the huge ecosystem which resides up to several kilometers below the earth crust is devoid of photosynthetically derived organic resources because either the sunlight nor the photosynthetically derived organic resources, organic compounds, are available in many of these deep crust systems. And accessing those deep crust environments are also very challenging, particularly if we take into account the chances of contamination, contamination with respect to when we try to access the deep subsurface environments, we often expose the subsurface environment to atmospheric air, water, or other factors. And overall, this earth crust biosphere remained less explored and mostly remained enigmatic. So, this deep biosphere is considered to be the largest microbiological habitat on the earth. As you can see, if we take a small portion of this crust, you will be able to find two distinct portions. One is the called oceanic crust, on which the oceans are placed, and the continental crust, on which the continents are located. Now, deep down, up to maybe 100 kilometer, we have the solid and rigid lithosphere, which is entirely made up of the rocks, often some sediments and other metamorphic materials are present. Now, up to 95% of the prokaryotic organisms like the bacteria and archaea, which amounting to an enormous number of cells, like in the order of 10 to the power 29 cells, resides in the deep subsurface on the earth. And this is so interesting that if we just look at this data on the right side, compared to the soil and aquatic ecosystems that we often see, and we consider them excellent habitat for studying microbiology, the terrestrial subsurface represents around 39% of the total prokaryotic numbers, whereas the marine subsurface consists of the 55%. Now, to take you further, now this deep history of earth biomass is very interesting. Now, ever since we realized that life is present within the deep earth crust, the deep biosphere, that is the biosphere which resides within the earth crust, has been recognized as an ancient, disparate, and diverse ecosystem of global biogeochemical significance. It is very significant considering the global carbon cycle in particular and the cycling of other nutrients in general. Now you can understand the significance of the carbon cycle and the nutrient cycle when we consider the climate change effect, carbon sequestration, capturing energy through biological or microbiological processes, or controlling the nutrient availability in our ecosystems like the productive ecosystems like the paddy or the agricultural fields. Now, apart from their importance in global biogeochemical processes, the deep subsurface also provides an analog for the habitats on Mars as well as extrasolar planets. So it has been identified in last couple of decades that the life which persists deep under the earth crust could provide us very valuable insights about possibility of life in extraplanetary or extraterrestrial habitat, particularly in Mars-like environments. Now, if we look at the data further, the subsurface deep biosphere represents 1/10 to 1/3 of the Earth's total global present day biomass. But if you consider the evolutionary background of these, before the evolution of the plants, these organisms, like the organisms which live under the deep continental crust or within the subsea floor environments, used to be the most predominant one, representing up to 80% of the total microbial populations present on the entire planet. And many of these organisms are still surviving deep under the earth crust and have been found to provide important information about the origin of life, evolution of life, and evolution of the entire planetary process, particularly the habitable conditions that we see today on our Earth, apart from the biotechnological and other applications that we can derive from these organisms. Now, recent estimates of the subsurface habitable zones, as you can see here, this is a very recent data which indicates that subsurface cells concentrations are determined across the different continental locations, and it is quite astonishing. Like in the continental subsurface, it could go up to billions of cells per cubic kilometer. And if we want to compare this number of cells or the total number of cells compare with the soil or the oceans, you can see that the underground, that is the underground crustal systems, not the oceanic one, but the continental crust could actually harbor more number of microbial prokaryotic cells compared to the existing or maybe the pre-existing belief that soil could be one of the richest microbial habitat or the oceans. Now, with respect to this subsurface, deep subsurface within the continental crusts, I will introduce you to two systems. One is very popular, the basaltic crust, another is the granitic crust. So these are very fundamental concepts about the knowledge of geology, which says that the basalts and granites both are basically igneous rocks, and the basalts are rocks which are formed because of the cooling of the magma, which is released following the volcanic eruptions. And across the Western Ghats, our famous Deccan traps, as you know, is the best examples of the basaltic lava flows. So these are basically lava flows which resulted from the very ancient volcanic eruptions, and following the solidification of those lava flows, the basaltic rocks are produced. Now, in contrast to that, the granitic rocks are intrusive igneous rocks. They are also formed from magma, but they are never exposed to the atmosphere, and they remain as a rock under the ground. Now, during our study, I will take you through the samples, which are both basaltic as well as granitic, and these are considered to be one of the most ancient type of rocks, because as you know that the planet has formed because of the consolidation or the crystallization of the magma from the earth core. Now, these rocks, these basalt and granite rocks, they constitute around 95% of the crust. And if we consider life within them, they are possibly representing the extreme habitats for life. Why they are considered extreme? Because they are within the aphotic zone, no sunlight, devoid of organic carbon, because naturally in these samples, the level of organic carbons are very low. The the contribution of photosynthetically derived organic carbon is negligible, and also the overall biomass production is slow. So the biomass which could otherwise be generated from microbes or algae or fungi are generally very poor. And interestingly, these rocks are subjected to high temperature and pressure at some point in their history. And overall, from a microbiological point of view, they are considered to be more oligotrophic, because they are generally having a very less concentration of different nutrients. Now, recent studies on deep biosphere is breaking the boundaries between the abiotic and biotic world of the geoscience. So it is emerging as one of the most intriguing interdisciplinary field within geoscience, because it hosts the huge catabolic repertoire. Considering the number of cells, considering the size of the crust, it's huge. And very interestingly, it remains largely unexplored biogeochemically and microbiologically as well. So these are the some of the locations which are indicated in this map, wherein the deep drilling sites are deep drilling is going on in order to access the samples. As you can understand, without drilling, we are unable to access the samples which are situated deep under the earth crust. Now, few important points. Now, organisms inhabiting these environments, likely to have isolated from the surface world for hundreds to millions of years, because they are sitting deep inside the earth crust, and they are often disconnected from the rest of the crust, which is the upper horizon of the crust. Their metabolic lifestyles may differ substantially from those of the surface organisms. And even though subsurface environments are diverse, these microbes often share common biological challenges, like the limitation of energy, resources, space, as well as different extremities like the pH extreme, temperature extreme, pressure extreme, osmolarity extreme, etc. Now, on the other hand, the subsurface environments offer biological advantages, because the environment is quite stable. It is not subjected to the perturbations which are often experienced by the surface ecosystems. They are protected from the lethal UV radiation and also the oxygen. These unique subsurface conditions lead to communities that are often phylogenetically and functionally very diverse. Now, how do we access these samples? Now, these samples from the deep crust can be accessed either through scientific drillings, where boreholes are drilled to collect the samples. Otherwise, we can have a number of alternative windows like different type of springs and wells, underground locations for waste disposal facilities, research facilities, and often the deep mines, because the deep mines are naturally dug to access the minerals and ores, and these tunnels are often used by scientists to access the subsurface environmental samples. Now, I will take you to the overview of the subsurface continental environments. So at the center, you see piece of the earth crust, where you can see this is the top surface, and these are the subsurface horizons. These horizons are not uniform. So maybe the initial part is the soil and followed by the vadose zone. And then you have the shallow aquifers and then the rocks, fractured rocks, and then more crystalline rocks as you go down. Now, in the upper horizons, like the soil and shallow horizons, the microorganisms generally live within the spaces like the water channels and within the space available through different grains. Now, deep inside, like hundreds or few hundreds of meters, since it is mostly hosted by rocks, and these rocks are often moderately crystalline. So within these rocks, we see that there are many fractures, and within these fractures, the microorganisms live, and they use the nutrients which are available following interactions with the rock minerals. Now, deeper down into the crystalline granitic or the basement rocks, where we have much less number of fractures, these organisms, number of organisms are also found to be or considered to be less, and still the microbes live inside the tiny pores or the fractures which are present within these rocks. Now it is very interesting that as we go down from the upper shallow aquifer or the vadose zone towards the more crystalline and more mm, solid basement rocks, the temperature increases. Temperature increases and also the pressure increases. And it is not only that the temperature and the pressure increases as we go down, but also the porosity of the environment decreases. Now, what is the role of porosity? More the porosity, more will be the movement of the organisms as well as the nutrients and the other chemical molecules. So less the porosity, that means the organisms will be not able to move, neither nor the nutrients etc will be able to move freely. So as we go down, along with the increase in temperature and pressure, the porosity of this crust decreases, making the life of these organisms more challenging. Now, there are two more important aspects. One aspect is close to the surface horizons. Photosynthetically derived organic carbon is available because photosynthesis is taking place on the top of the surface, and these photosynthetically derived carbon can penetrate into the underground system, and they can diffuse up to some extent and then provide some resources. But the extent of that declines as we go down. Nonetheless, in the deeper horizon, we see the life could be fueled or supported by different type of geogases. So these are purely geogenic gases originating from magma or from different rock water interaction and other abiotic reactions. These gases and other nutrients are produced. Now, we must know that when we try to investigate microbiological processes within any geological environment, we consider it as a subject of geomicrobiology. And these geomicrobiological processes, they occur with respect to three vectors. One of the vectors could be surely the catabolic landscape. Like in the catabolic landscape, we consider the species composition, like the microbes which are present. They are the genomic content, the enzymatic repertoire, their ability to interact with themselves and ability to interact with the inorganic and organic nutrients. But also, these two other components are very important. One of them is the chemical landscape, which represents the nutrients to be, like the compounds which can be utilized as nutrients by the microorganisms, including the electron donor, electron acceptor, carbon source, and different stressors, as well as the abiotic landscape, like the humidity, conductivity, temperature, etc., which controls the processes. Now, altogether, the geomicrobiological processes is subjected to all three controls. Control 1 is the microbes, control 2 and 3 are the chemical and the abiotic processes which are taking place there. Now, with respect to the deep subsurface, as I mentioned that earlier, that the available energy resources could control the structure and the function of the crust, because with the photosynthetically derived organic carbon diffusing underground through the rainwater during the groundwater recharge system, the shallow horizons, including the shallow aquifers and the deep aquifers, they are often fed by the photosynthetically derived organic carbon. They could be transported well up to certain depths. But this could supply actually some organic carbon from the ground surface up to some levels within the hard rocks. And that could actually result into a predictable sequence of terminal accepting events like aerobic to anaerobic respiration and then methanogenesis, as we often study, because of the flow of this organic carbon from the upper horizon can facilitate this process very well. But as we go down, this flow of organic carbon diminishes, because many a time, because of the diffusional barrier and the concentration and utilization of the organic carbons, the organic carbon remains unavailable to the lower horizons. And therefore, in the lower horizons, in general, we see a much less amount of cellular activity in terms of carbon dioxide production. However, what is most interesting is this, that although the organic carbon is deprived, however, these deeper horizons could be fueled by different geogases. Geogases like the hydrogen, carbon dioxide, methane, etc. Like these gases, hydrogen and carbon dioxide, which could be originating from magma, could be rising from the serpentinization, radiolysis of water, or oxidation of different silicate minerals and mineral dissolution. So ultimately, it's the carbon dioxide and hydrogen which are originating from the different abiological processes could actually facilitate a large number of reactions. And these reactions could be organized into structure like this, that these could actually allow acetogens, acetoclastic methanogens, and autotrophic methanogens to produce different organic polymers, and these organic polymers, organic substrates can be oxidized and they can produce subsequent precursor molecules and other metabolites on which other microorganisms can survive. Now, based on that concept, hydrogen driven life was proposed, and it was found to be successful, at least at some of the deep subsurface environments. The life is completely driven by hydrogen, and this hydrogen is originated from the abiotic reactions, particularly from the interactions between the rocks, the minerals with water, and other abiotic factors. Now, given the extent of the deep biosphere, a series of wide-ranging research questions remain unanswered. Although scientists are investigating deep biosphere for at least 30 years or so now, but still some questions remain unanswered, like how deeply does life exist into the earth? What fuels the deep subsurface microbial life? And how does the interplay of biology and geology shape the subsurface? And what the subsurface genomes could tell us, and how life on the surface originate in the underground system, and these could also culminate into certain we call decadal goals to determine the process that define the diversity, determine the environmental limits of life, and determine the interactions between the life and the carbon cycling on the earth. From evolutionary perspective to the biotechnology or the climate change mitigations, all these questions are found to be highly intriguing and important. Now, what remained largely unexplored is the distribution and the diversity of microbes in terrestrial deep granitic crust in particular. Because in the oceanic crust, still there are some expeditions that is called under the purview of the ocean drilling programs for last 20 years or so. Several oceans have been mapped, and the under the ocean environment, particularly the sediment, deep sea sediments have been investigated. Compared to that, the terrestrial deep granitic crust remain almost understudied. And knowledge on the metabolic functions of the organisms which might be living in this crust and their impact on global carbon and nutrient cycles remain unexplored. And also, when we look into the different type of geologic processes like tectonic processes or seismic activities, we don't have any idea about how these organisms, etc., could be linking to. Now, these igneous environments or the crustal environments are basically studied under two contexts. One is the terrestrial basalts and the marine basalt, as I mentioned earlier. So these are the different geological locations so where these studies have been conducted. And in general, considering the accessibility of the samples, the water samples which are obtained from the aquifers or the faults and fractures present in the underground environment are studied because water can be extracted relatively simply. Endolithic rock microbiome from the continental crust remain unexplored. One of the reasons ascribed is the difficulty in accessing such samples because it's not water that you can put a pump and suck the water out. If you want to take out rock sample, you need to use certain instrument, you need to drill, or you need to use certain other processes. And the chances of contamination and scientific endeavor is very much important for those things. Now, with respect to that, Government of India, the Ministry of Art Sciences, has taken up this particular initiative of scientific drilling in Koyna. Now, I will introduce with this name Koyna. Koyna is the name of the river, but Koyna is also considered to be a very important region in the Western Ghats because of the impoundment of, as you can see in the map, it's all the Western Ghats. And the Koyna Reservoir was impounded in 1962. It's a big dam producing electricity. But soon after the impoundment of the dam, major earthquakes happened. And it is identified that till date, there are numerous such earthquakes, close to several thousand earthquakes are happening. Every day earthquakes are happening. All earthquakes are not that significant. And why earthquake is happening in that region? Because of the loading of the dam. The water gives pressure to the underground crust, and as a result of that, the crust is developing some kind of stress. And the stress is released through these seismic activities or the earthquake triggers. So Ministry of Earth Science, they have planned to drill a number of exploratory boreholes. The black dots are the exploratory boreholes. Exploratory boreholes are up to 1,500 meter or so deep to investigate the seismic process and also to investigate the geophysical properties of those regions because of this earthquake prone nature of this environment. And also, the extent of the earthquake is actually highly controlled by the, the discharge or the recharge of the dam. When the dam is receiving water and when the dam is discharging water, during these two times, the earthquakes are mostly happening. So in other way, this region represents a kind of a man-made huge laboratory where natural systems can be investigated. Now, you may be thinking that what could be the role of a microbiologist in this system? Yes. So that is the question, that what microorganisms could do under this kind of system where deep earth crust is subjected to seismic activity. Now, well, for that purpose, I need to tell you something, that when these drill holes are made, I will show you some pictures later. The drilling process is a scientific process which allows us to get the rock samples from the underground under scientifically appropriate condition. So earlier, as I was mentioning, that getting the samples from the deep crust is very difficult and challenging. It's expensive and it's scientifically very challenging. Not that everyone can take a drill and go there. It's a hugely expensive and complex process. Now, since the ministry is already establishing these borehole laboratories, each of them are instrumented. So they are considered to be small laboratories, each of them. So we are able to access the samples. And also we are able to study some of the most unique habitats within the world, because this is the part of the Deccan traps. So I will come to the Deccan traps later, that why Deccan traps itself is so important. So as you can see, there are multiple locations through which the samples were obtained. And also in the later stage, a deeper borehole, which is 3,000 meter or 3-kilometer deep borehole, is drilled at this position. And these are all close to the epicenter of the major seismic activity. Now, this is the picture of the main drilling site. It is taken by a drone, as you can see, and you can see also the terrain. In the top, it's the top of a mountain actually. So this is the top of a mountain, and we have flattened this and established this huge facility to drill. And this is the drill rig. This is not a village or township. This is only for drilling this deep subsurface. This establishment was created, and these are all the quarters for the scientists and staff, and these are the instrumentations for the drilling process. So this is the drilling rig through which 3,000-meter deep borehole was drilled and sampled. Now, as you can see in this particular cartoon, we are trying to analyze the deep subsurface earth crust microbial systems, but we are also having opportunities to study two different horizons. What are these horizons? If you look carefully, this part, which is marked as Deccan basalt lava flows, this is the huge pile of Deccan basalt, which is globally important as and named as Deccan traps. This is around 65 million years old. So 1 million years means 10 lakh years. So you can imagine that 650 lakhs years old. This is this. And these colors indicate that it has undergone several rounds of volcanic lava flows. So it's not that huge, several kilometer deep rocks of basalts are not produced in one day. It took around 10 million years to form this Deccan basalt. So when we are drilling through different exploratory boreholes, we are able to access the rocks which remain underground. And generally, these rocks are devoid of any aquifers or anything. In the shallow horizon, there are some aquifers, that means groundwater, but the deep inside, there are not much of water. So they remain almost protected from the external environment because water is not penetrating there. These are very solid rocks. And so now you can imagine that these are 650 lakhs years old rocks, remain unexposed to external environment for a very, very long period of time. And more to this, these rocks are situating or sitting over the basement rock. This is called the basement. This basement is called Archean basement. Now, what is Archean basement? That means it is nearly 2.5 to 3 billion years old. So ideally, you can imagine that during or before the volcanic eruptions of the Deccan volcanism, that is 650 lakhs years ago, dense forest might have been there at this region, like in the basement rock, and many animals, including the dinosaurs and all these animals, might be there. And then that volcanism happened, which is also known as the Great Extinction Event. So everything was ashed or burned totally, and that continued for 10 million or 15 million years. So interestingly, we have another system now, which is the basement. Now, this basement is more primitive, close to the origin of the planet or the origin of the lifetime, and it remains protected from the external environment further because of the blanket of this thick lava pile. So the lava pile itself is one layer, and beneath that we have the alkine basement rocks. So through the exploratory boreholes, first we sampled the basalt samples, and then through the pilot boreholes, what picture you can see over here of the drilling rig, we were able to sample the rock cores up to 2,908 meter deep inside the crust, and we investigated the life present in that environment. Okay, so the questions that we asked are very simple: who they are, what are they doing, and how they are involved in the evolution of the process, the evolution of this geobiosphere. So we took the samples. So I will take you to the details of this. That we took the samples, which are rocks. We powdered the rocks, extracted the DNA, and analyzed it. So here we have used some basic philosophy of biodiversity studies. Some of you or many of you might be knowing this concept. That we believe that the samples are multi-species entity. So rocks could be multi-species entity, and you can isolate pure culture and extract the genomic DNA and go for 16S ribosomal RNA gene analysis, or you can enrich certain populations and analyze that mixed DNA, or you can extract the metagenomic DNA and go for 16S rRNA gene analysis, or you can go for whole metagenome analysis. Now, when we go for the 16S analysis through a next generation sequencing based technique, we prepare the genetic soup, which is basically the extracted genetic material from the rocks, and then we amplify the 16S and we get the reads, and these reads are analyzed to produce the microbial community composition. In specifically, we are going to answer the microorganisms like bacteria, archaea, eukarya, who are there, what are their metabolic potential, how are they interact, and how they respond to the external perturbations. Now, some general description about the OTU pickings, operational taxonomic units. I think many of you already know that we consider them as equivalent or closely equivalent to unique species, but not exactly the species. So these are based on 97% or 99% identity. These reads are clustered and OTUs are formed, and the OTUs are subjected to taxonomic identities. Now, during the whole metagenome analysis, we do not use any PCR amplification of any genes. So for 16S, we use PCR, but for the whole metagenome, we do not use any PCR. Rather, we just take the DNA of the genetic soup and fragment it into millions of short pieces. And these short pieces are sequenced through NGS, and then the reads are generated, and these reads are analyzed through assembly and annotation pipelines to get the interpretations out of the data. Now we coin a pilot borehole, and as I was showing you the picture, and in the phase one, the samples recovered to exploratory boreholes, which were shallower, up to 1,500 or 1,600 meter deep, and mostly they provide us the sample from the basaltic and few granitic horizons. So these are the rock samples, as you can see, and we are able to extract the rock powder, and from the rock powder, we extracted the DNA. Now, if you look at the geochemical data, we see that the geochemically, these rocks are quite distinct. Like you can see that the basaltic horizons and the granitic horizons are absolutely distinguished. from the geological point of view. So there are many parameters that we were estimated, and based on those parameters, we found that geologically these rocks are chemically also, geologically means geochemically also, these rocks are different. So if I want to tell you very simply that we are analyzing this basalt, which is 65 million years old, and then we have the granite, which is around a few billion years old rocks. And these rocks are protected from external disturbance or perturbations by these basaltic rocks. So when we analyze these basaltic rocks, mostly the basaltic rocks through this 16S rRNA gene amplicon sequencing, you can see that with increasing depth, the data about this microbial community composition. Now, the communities are found to be highly constrained by the geochemistry of the rocks. Now, the basalt, the granites, and the transition zones. These horizons, they harbor unique microbial groups. And these microbial groups are constrained or controlled by the geochemical conditions, which is very rather simple to understand, because the microbes are governed by the prevailing geochemical and geophysical conditions. But it is very important for you to understand that when we look at the community composition, apparently the difference is not so clear. But when we use the statistical tools like a Bracardis analysis or so, we are able to discriminate the samples based on their microbiological as well as the geochemical roles. We can define that which geochemical parameters are actually controlling what type of microbial activities. And then we built the co-occurrence networks, and we identified that both fermentative as well as anaerobic syntrophic organisms, organisms involved in syntrophism, are involved in interacting with themselves, and they are also subjected to a basic whole metagenomic analysis to understand the carbon, nitrogen, and sulfur cycling gene. And we are able to identify the major carbon fixation or the nitrogen assimilation or the sulfur metabolism pathways. And overall, from this primary investigation, we were able to delineate the major biogeochemical cycles which are relevant for these relatively shallower horizons of the crust, which is not actually shallow in a general sense, but to us it is shallow because further we have gone down up to 3,000 meters. So now the basement granitic samples. So followed by the initial samples on the initial investigation on the basaltic rocks, we now took the granitic cores. As I mentioned, these rocks are more ancient, billion years old rocks, and perhaps they were never exposed to outside environments in the geologic past. So as you can see, these are the depth of the samples, and the temperature of these samples are also increasing. So up to 73.9 or 74 degrees. So the lowest, deepest sample is around 74 degrees centigrade. And we were able to successfully extract the metagenomic DNA, and we analyzed the 16S rRNA genes using multiple replicates. And I will show you some interesting findings of this. Now you can see that we try to gain a huge amount of data out of this. This is 10 to the power 6. So we got the data in the order of millions of reads from each of the samples. And when these reads were clustered, you can see that the number of OTUs. And it is in the order of 10 to the power 4. That means if we consider each of these OTUs are equivalent to a species, then that means there are actually more than 4 into 36 10 to the power 4 species. So this is significant that in spite of such depth, such extreme conditions, the number of species are truly very high. And with depth, because these are the deepest samples, the number of OTUs are slightly less, although it's not that significant. Now, when we try to determine the taxonomic identity of these organisms, like the number of genera present in these samples, it is Very interesting to note that with the depth, the number of genera are actually increasing. So it was surprising to us as well because the deeper horizons, as I mentioned, are more extreme. So they are. It is not that the extreme conditions are not inhabited by organisms. In fact, the extreme habitats are found to be more populated in terms of the different the bacterial genera present in that. Now, this is the microbial community composition. As you can see, the community is mainly composed of some taxa, but and it is overall presenting a common trend, except the the intermediate depth region, where we see the increase of some organisms, including the methane oxidizing verrucomicrobia or some cyanobacteria, are very distinctly detected in some intermediate horizons. And what we also observed that within these granitic horizons, if we took this microbial community composition data, the microbial communities are constrained by the depth. Like you can see that this analysis suggests that the samples are arranged according to their depth. So the microbial communities are arranged or composed of organisms in such a way that the depth has a very significant role on that. Now, microbes in each of these horizons are interacting with themselves. So, in order to study these interactions, we use the Spearman correlation. So, for each of the samples, we have multiple replicates. So, we use the Spearman correlation, and we observed that the correlation pattern changes with depth because more red means that is where the correlation is higher. Now, if I take you the overall pattern of co-occurrence between all the samples taken together, we'll see, if a close-up view, that organisms who are most interactive are very interesting type, like Dinococci. Many of you might know that Dinococcus is already renowned for its extreme desiccation and radiation tolerance. Cyanobacteria, who knows what cyanobacteria is doing there in this deep earth crust. Then anaerobic, anaerolini, thermoleophilia, and many other organisms. So when we try to look into the metabolic potential from the literature, we observe that this is actually an extremophile snook because the environment is highly desiccated, no water, high temperature, high pressure, but the organisms are also falling in line with that because most of these organisms which are most interrelated are previously known to be desiccation resistant, temperature resistant, radiation, and other stress resistant type. Now, in the next, I will show you another important aspect, which is analyzing the core community or core microbiome. What is core microbiome? Core microbiome is comprised of the members common to two or more microbial assemblages. That means organisms which are present in all the samples represent the core members or the endemic members. And these endemic members or the core members, that means they are common to all the samples, they seems to play key roles in the ecosystem function because with change in samples, these organisms are not changing. Their relative abundance might be changing, but they are not disappearing. So we try to identify this, and what we found that across these granitic horizons, although we have a huge number of OTUs from each of the samples, a handful of OTUs, like 5,724 OTUs, remain shared across the samples. That means these OTUs are conserved across all the samples. And these OTUs alone, they represent up to 60% or 61% of, or 62% rather, of the community. That means a handful of species are shared among all the samples, and these samples, these members, these microbial members, they represent a significant fraction, a portion of the total community. Now we determine the taxonomic composition, and we again found that these members are mostly affiliated to the members which are previously detected, but a distinct pattern was observed. Like with respect to depth, if you consider the some members like Verocomicrobia or cyanobacteria, etc., they varied significantly, whereas some members remain quite consistent. Now, with respect to the core community, because who controls this core community? Now we found that core community in fact is having two types of distributions. In the deeper horizons, the core communities are more constrained by hydrogen, methane, alkalinity, temperature, and lack of TOC or the total organic carbon. Compared to the shallower samples, shallower means the samples which are from relatively upper horizons. Now, we built the network among these the most important core community members. Now, what we found that these networks are actually showing us that very interesting assemblage of two types of organisms at least. One of the organisms are the member of the actinobacteria, which are known to be extremely stress resistant, and another type of organisms are the heterotrophic and autotrophic proteobacteria and firmicutes members. So our later investigations actually provided more insights that how these processes could be explained or interactions could be explained. Now, if I take you to this network, which is a kind of another representation of the same network, you can see the members of actinobacteria are most connected. Now, what are these actinobacteria? These actinobacteria are most stress resistant. They can actually resist multiple stress, and then almost in all cases, like in Atacama or desert to Death Valley to multiple extreme habitats, these organisms are reported, and they also try to engage into different type of metabolic interactions with multiple organisms, and along with them, the firmicutes are also there. Now, we also observe that it is not that the networking is simple. With respect to depth, if we consider, we find that some organisms correlate very well with the depth, and some of the species actually correlate very well with hydrogen or even methane also. As you can see, with respect to hydrogen, the interaction is more robust, and some of the organisms are very clearly interacting with hydrogen. So that means this kind of network analysis helped us to understand that how hydrogen, which is considered to be one of the major fuel for this environment, is actually allowing some organisms to utilize it very effectively and then providing the resources for other microorganisms as well. Now, at the end, we performed the whole metagenome analysis. So we took the entire DNA, we fragmented it, and following fragmentation, we sequenced and performed the assembly and annotation, and the metabolic pathways were reconstructed. So what we observed from this metabolic reconstruction, this is a summary. On the left side, you see the genes, different genes for the carbon and energy metabolism. And these genes are normalized with respect to a taxonomic marker, that is RPOB gene. And on the right side, you have the taxonomic affiliation of these genes. What is very interesting over here is the abundance of this gene, RNF complex and the HOX. Now, what are these RNF complex and HOX? These are the genes which are involved in oxidation of hydrogen or use of hydrogen. And also, the genes like ACSS. This ACSS is the gene which is responsible for metabolizing acetate into acetyl-CoA. Now, you might be knowing that acetyl-CoA is a very important product of the glycolysis and the TCA cycle. So we get the pyruvic acid from the glycolysis pathway, and the pyruvic acid is oxidized further to produce the acetyl-CoA, and acetyl-CoA is utilized into the TCA cycle. Now, interconversion of this acetyl-CoA to acetate gives a kind of a very unique opportunity for these organisms in this environment that we are investigating now, and which is called acetate switch, because that acetate switch is considered to be involved in controlling the stress response and the overall metabolic behavior of the community, particularly when it is under stress conditions. Now, based on this process, we are able to define certain basic metabolic reactions, as well as the basic microbial groups which are involved in such processes, which are under the final level of validation. Uh, this particular set of information. So at the end, we are trying to investigate this engine of the deep biosphere, which seems to be driven by the oxidation of hydrogen, coupled with fermentation by the heterotrophs and an oxygenic CO2 fixation. That is basically the Wood-Lung-Jal pathway, and coupled with acetogenesis, which is going on, and that is driven by the geogases like carbon dioxide, hydrogen, and methane, etc. So, what drives the deep aphotic life? So, to our understanding, it is the hydrogen and carbon dioxide, because the CO2 fixing bacteria, diverse firmicutes members, particularly, they are able to run this Wood-Lung-Jal pathway, produce the organic polymer, which is used by fermentative bacteria and also used by iron sulfate reducing bacteria, and they are producing the acetate. And then the acetate is undergoing this acetate switch cycle, and acetate is allowing the methane formation by acetoplastic methanogens, and also the methane could be also produced by hydrogenotrophic methanogens as well. And the overall biomass which is being produced is continuously being recycled, as there are methane oxidizing bacteria also. Overall, in summary, this study is exploring the geomicrobiology of deep subsurface of the Deccan Traps and underneath granitic basement through molecular genomic approaches. And distinct geochemical conditions are found to prevail in the subsurface origins. Low organic carbon, moderate sulfate, iron, nitrate, etc. allow specific microbial populations to flourish, and they demonstrate a niche partitioning within these habitats. And the lack of water, other mechanism of cell migration potentially allowed deep communities represented by thermophilic anaerobic microbes capable of sulfur, hydrogen, and methane metabolism. And the distinct role of autotrophic and heterotrophic microbes in regulating the subsurface biogeochemical cycles were elucidated. So at the end, I thankfully acknowledge Ministry of Earth Sciences, Government of India for providing all the supports. Borehole Geophysical Laboratory at Karad for providing logistics and intercontinental drilling program, a deep carbon observatory for supporting our work, and all the drilling teams, team members, and all the fellow students. So these are the drilling, some of the drilling crew and staffs over the drilling site, and the drilling rig on the background. Thank you so much for your time and patient hearing.
Thank you very much, sir.
Thank you.
Um, I would like to ask the audience if they have any questions. Yes, Dr. Jyoti.
Oh, hello, Dr. Pinaki.
Hello, Dr.
Yeah. Hello, sir. Very intriguing study. I just wanted to ask you that you have been, you have shown one particular region in the that is your C8 component, okay, which is pretty close to the crust of the earth, right? The C8 component where you did the drilling, the C8 core. And so, did you find any kind of overlap between the organisms that you obtained in your ballast region, that where the the larvae etc. they have been deposited over the years, and the C8 component? Because what you would expect is this the when the volcanic eruptions are taking place, they would be carrying the microbiome from the crust region to the top, and they would be depositing it in those rocks at the top. So did you find any kind of an overlap between them?
No, so thank you for your question. Actually, there is a point. There is no microbes in the lava, because the lava must be of very high temperature.
Okay.
Okay, so don't expect microbes to be that smart to survive few thousand degrees centigrade temperature. They cannot. Number two, these rocks are actually consolidated crystalline rocks. So the basaltic rock is formed when it is exposed to atmosphere, and the magma which is thrown out as the volcanic eruption get exposed to the atmosphere, and slowly over a few years time it solidifies and forms the rock which is called basalt.
Okay.
So it is not expected that the basalt and granite will have similar bacteria. Some bacteria may be similar, but in its characteristic type, the basalt is different from granite because granite is intrusive. It is not exposed to atmosphere. So atmospheric reactions like exposure to oxygen particularly are not there. So the mineral composition, the elemental compositions, before microbes start living on that, are different. Okay, so you don't expect that C8 and the basalt would have uh, much of overlapping. However, because it is igneous rock, overall it is igneous rock. All basalt granites are igneous rock. So some organisms are omnipresent.
Okay.
Those organisms are expected. But
Okay.
anyway, they are two different habitats altogether.
So basically, the basalt, I mean, microbiome would represent what has developed after, all the rocks have solidified. And
Of
yeah, yeah.
course. Because in a very hot condition,
Mm-hmm.
some spores might survive, but you can imagine a volcanic lava flow,
Yeah.
and readily it will not allow any life to sustain. But
Right, right.
over some period of time, microbes will occupy those habitats as
Mhm.
they become habitable. Like temperature comes down to around 100 degrees centigrade or so.
Okay.
Slowly, microbes will start colonizing. So
Okay.
that might have happened. We don't have any idea because there is no fossil.
Okay.
Microbial fossils are not detected in these regions. So we
Mhm.
don't have any fossil record. So we
Okay.
only have the DNA and a few living bacteria. That's all.
Okay. Okay. Thank you so much.
Thank you.
Yes, Dr. Vinit Kumar, please do
Yeah,
ask your question.
sir, good morning. It is very interesting to hear your talk. It is very informative also. Now I understood why all the international scientists or NASA is looking for life anywhere in the other planets. So by doing the core and all. But thing is, I just wanted to know whether whether you have you any find this live organisms in the granite core, or only DNA you find?
Okay. So thank you, Dr. Vinit Kumar, for your question. Yes, we could actually retrieve living organisms, living bacteria from almost all the samples.
In that, you find a lot of actinobacteria,
Mm-hmm.
actinobacteria, and many other bacteria. It is very interesting to note that whether it is these bacteria are culturable bacteria, or only they are we can extract through DNA.
Okay, so that's a very interesting point. Most of these organisms are not culturable. Okay,
Yeah.
and only when we provide some specific
Sir,
substrates.
Dr. Pinaki sir, just wait. Dr. Irene,
Yes,
मैडम
yes.
मैडम आप माइक म्यूट कर दीजिए थोड़ा.
Okay, okay.
Thank you.
Yes.
Sorry for that.
Thank you, Dr. Garg. So what I was saying that most of them are not uncultivable, as you said rightly, Dr. Vinit Kumar, and we were able to revive, I will say, revive some of the organisms as laboratory culture only when we provided with some specific substrates like hydrogen.
Mm.
Okay,
Yeah.
and of course, to add to more, isolated pure cultures
It's very
are
important.
are very difficult to obtain. Okay, So most of the time they grow as enrichment mixed culture. Okay, as you know, these are called enrichment culture because they may be growing on hydrogen or methane, high temperature, around 50 to 70 degrees centigrade incubation, and most of them are mixed. If you try to isolate them singly, single colony will never appear.
Yeah, okay.
So we are trying to work on them. So it will take maybe a few years more to get them investigated thoroughly.
Yeah, thank you very much, Professor.
Thank you. Thank you very much.
Hello. So just one more question. Is it possible, like, you when you're culturing this, and if you were to include a kind of a mineral extract or something of the rocks in your media, would you think something like that would help? Of that medium? Just a thought.
No, you are asking something as if you have worked in my lab itself, or we are collaborating. Yes, you are right. You are absolutely right. The medium that we are using is a special medium that we have prepared, and it is actually not like a soil extract medium, So we do not extract the mineral composition, but we try to replicate them. Okay, because these minerals are insoluble, so we rather use the soluble salt of these compounds, and we found that yes, indeed that helps. If you add those compositions which are similar to exactly the mineral components, it helps a lot.
Okay.
It helps a lot.
Are there any further questions? I have a question.
Yes, please go ahead.
If there have been any whole microbiome studies done on the deep subsurface ecosystem, have you all found viral sequences in those?
Okay. We haven't actually mined the viral sequences yet. Maybe we are planning, or we should be planning that, because our primary aim was bacteria. But you are right that in other locations, like in South Africa particularly, and in some European deep subsurface, people are observing that there is a significant presence of viruses. Okay, that there is virus. So we haven't studied because one reason is that it was not included in our primary objectives, and also the amount of the reads that we received were not that significant in order to cure it for viral or virome study. Okay, because most of the 99.9% reads belong to bacteria or archaea. So very less number of reads are available for anything else like eukaryotes or virus even. So some specific study may be done, but there are reports.
Thank you, sir.
Thank you.
If there are no more questions, can I propose the vote of thanks?
Can I ask something? Dr.
Yes.
Judith?
Yes, Dr. Irene, please go ahead.
Thank you, Professor Pinaki, for your such a lucid and such a informative, especially your last slide, running the engine, was so interesting.
Thank you.
But a few thoughts on my side. Would you consider acetoclastic switch as an important driver in this microbiome?
Yes, it is acetate switch as people have defined it earlier. Yes. Actually, ma'am, what we observed that this acetate switch is not a very regularly observed phenomenon in such extreme habitats. Okay, so it is observed in certain other habitats, but it is a very interesting component to control the stress management, because how the metabolic flux of the acetate or acetyl CoA is divided between growth metabolism or sustenance metabolism.
Yes.
So because it is very clearly reported in a recent Japanese studies that they have done some single cell genomics to identify that this acetate switch actually controls the carbon flux either towards growth metabolism or to stress metabolism. So with respect to this extreme microbiome, it would be of great importance and significance for us. So we are looking forward to investigate that part in future. But surely you are absolutely right. As acetate switch is a very important point in that.
Yes. And my second, thought is what do you attribute to finding cyanobacteria there? Because it is a photosynthetic probacteria.
Yes. So, like you, I was also very surprised when we initially got cyanobacteria. Then, after some literature mining, because that time we were not having the metagenome data, only the 16S data, we could find that these cyanobacteria are evolutionary very important. Because what we perceive cyanobacteria today was not originally that. Cyanobacteria is involved in, you might know the event which is called great oxidation event. In a geologic past time like nearly 2.6 billion years ago, the oxygen evolving photosynthesis mechanism, which was not originally photo-driven but chemo-driven, was started in cyanobacteria. Okay, so in between somehow we forgot that process. We means general microbiologist. We some regular classes we do not talk about these things much. We think cyanobacteria are oxygenic, cyanobacteria are photosynthetic. See, but to the contrary to that, originally cyanobacteria could be actually not photosynthetic at all. They are utilizing chemical energy like oxidizing sulfur or oxidizing ammonia or nitrite, or they are actually oxidizing even hydrogen. Some cyanobacteria that we are getting are found to be fermenting also, and they are producing hydrogen as well. So unless we sequence their genomes, it will be hard to implicate and through a maybe transcriptomic study that how their genome is basically controlled. Because they oxidize hydrogen, they produce hydrogen. And they also oxidize sulfides. They oxidize nitrite, but they don't use light. Okay, they don't use light, they don't produce oxygen. So it's a very fascinating type of cyanobacteria, which I would like to talk to detail on that maybe later time, not today. These are connected to the evolution of life on the planet. Okay, a specific group is called oxyphotobacteria. These organisms are implicated in the origin of life, and NASA is currently looking into those organisms as a potent maybe proxy for Mars study as well.
Thank you, sir. And my last thought, which is maybe a foolish thought, but I just leave it to you. All life passes away, dies. So could it be that some of these bacteria which are there, or microbes which are there, they themselves die and supply the required carbon?
Yes, yes. And that is if you look at my what drives the deep subcycle, after that, what drives the deep aphotic life, these dead cells are providing, in fact, there are actually specialist group of microbes who are champion in utilizing the cell wall and cell membrane residues which are deriving out of dead cells, which we call necromass.
Yes,
so the necromass utilizing enzymes are abundant in deep subsurface, this kind of environment, because they don't leave anything. They just don't leave the hydrogen, they don't leave carbon dioxide, even they don't leave the dead bodies also of their organisms. So the dead cells are metabolized by different enzymes, and they produce precursors and metabolites for others.
Thank you very much, sir. Thank you.
Thank you very much.
Before we close the session, I would like to thank Professor Pinaki Sar for that very fascinating talk on a branch of microbiology that most of us present might probably have heard very little about previously.
Thank you, Dr. Judith, and all the team from the Department of Microbiology, Professor Gorg, and all the distinguished faculty colleagues and the participants, and all the members of this seminar. Thank you.
Yes, sir. Sir, I'll just thank you a little further. So, sir, just briefly mentioning some of the important points that you covered. So, you rightly said that microbes run the world, and for me at least, it was very surprising to know that the terrestrial and marine subsurface are far richer in microbial diversity than surface ecosystems. You explained to us the biotic and abiotic characteristics of the deep subsurface, specifically the Koina Reservoir, and it was very fascinating to know that there is
Hello.
rich diversity of microbial life
Acha,
in
acha.
these unexplored territories with an ancient history. So finally, we are very grateful that you could make it here today in spite of having a health issue. We truly appreciate your time. Thank you very much, sir.
Thank you. Thank you very much, Dr. Judith. Thank you, everyone, for your patient hearing and your time. Thank you.
Dr. Jyoti, could you please mute your mic?
So, can I leave now, Dr. Judith?
Yes, sir. Yes, sir.
Okay. Thank you, everyone. Have a nice day.
It can be degraded and its substitutes like substituted
Dr. Jyoti, could you please mute your mic?
various different like methods via various pathways.
Okay.
So over there, like
Professor
with
Keshav Prasad, we welcome you to the session.
UV
We will take a short
Great.
break
Chlorine
of about 5 to 7 minutes and start at 11:35.
तुम वो जो pathway जो explain करोगे,
Participants,
तो you
please
will just
stay
see
connected.
transfer
We will start
and
exactly at
secreted via,
11:35.
various different metabolic, like networks or pathways which do
Dr.
interconnect
Judith?
at some point or the other.
Jyoti.
Okay.
Dr. Jyoti?
So that way you can see, and then naphthalene का तुम दिखाओगे ये naphthalene pathway the
Dr. Jyoti,
first
can you hear
enzyme
me?
that is involved in this. So we have selected this particular
Yeah,
enzyme.
just go.
Salicylic. It reduces it to produce
Trupti,
salicylates.
if we can have her number, we can
Then
just call
salicylate
her.
is converted to genticillate or catechol via different pathways. So we have selected out of these two different enzymes. One is vanillin deoxygenase and salicylate
Hello,
monooxygenase.
Trupti.
And then you can say another enzyme
Just see if we can call Dr. Jyoti.
degradation.
Yes, sir. Trupti is calling her.
वैसे करके तुम वो पाठ्य का एक्सप्लेन करोगे पहले तुम पूरा ये जो तुमने स्लाइड बनाई है इंट्रोडक्शन की ओके सो दैट यू हैव सेलेक्टेड हाँ हाँ सो दीज टू पाथ हाँ तो वो जो पाथवे हाँ जो पूरा पाथवे एक ही में डालो ठीक है और उसको वहाँ पर वो एक्सप्लेन कर दो कि these are the three enzymes that we have chosen act at this point in the pathway, okay? And फिर जो तुम
Judith, okay.
हाँ, sir. We shall proceed for the next session now. Professor Keshav Prasad, shall we start?
Yes.
Okay. This session will be coordinated by Dr. Lata Gaude. Over to you, Dr. Lata.
Very good morning to one and all. A very warm welcome, sir, to Professor Keshav Prasad, who is our next speaker of today's session, and none other than our very own alumnus of Goa University, Professor T.S. Keshav Prasad, Professor and Deputy Director at Center for Systems Biology and Molecular Medicine at Yanapoya Research Center, Yanapoya, deemed to be University, Mangalore, since 2015. On behalf of our entire team of this 3 days national webinar, Technological Advances in Applied Microbiology, I, Dr. Lata Gaude, Assistant Professor at Department of Microbiology, Goa University, welcomes you, sir. And it's a proud moment to know that Sir is an alumnus of Goa University and obtained his PhD degree in botany, mycology from Goa University in the year 2004. Before moving to Yanapoya, Sir has been a faculty scientist at Institute of Bioinformatics, Bangalore from 2004 to 2017, an officer in charge of the Proteomics and Bioinformatics Laboratory at the National Institute of Mental Health and Neurosciences, Bangalore from 2012 to 2017. Sir has successfully established advanced proteomics and bioinformatics facilities and research teams at the National Institute of Mental Health and Neurosciences, Bangalore, and Yanapoya, deemed to be University in Mangalore. Sir has published over 250 scientific articles in the journals of international repute, including Nature Biotechnology, Nature Communications, Genome Biology, Genome Research, Journal of Proteomics, Journal of Proteome Research, to name few. And Sir is a recipient of the Young Investigator Award from the Department of Biotechnology, Government of India in the year 2007. Sir has an H index of 44 with a total citation of 13,448 and a cumulative impact factor of 1097. Sir was leading the efforts on the development of the Human Protein Reference Database, which is cited in over 4,000 international publications. And Sir's two papers, that is one which is Human Protein Reference Database, published in the year 2009, is ranked second. And the second paper, a draft map of human proteome, featured on the cover page of Nature in the year 2014, is ranked sixth among the top 10 publications with Indian affiliation in the area of biology and biochemistry, as evaluated by the Department of Science and Technology, Government of India, with the help of two independent, well-known international entities, that is Thomson Reuters and elsewhere, and published in the India's Research Landscape, Output, Collaboration and Comparative Performance, Bibliometric Studies on February 17, 2016. Sir was also involved in the development of the plasma proteome database and the database of human signaling pathways, NetPath. Presently, SIR's research group is focusing on genomic, proteomic, and metabolomic approaches to investigate infectious diseases and neurological diseases. For this, SIR is collaborating with a number of clinicians and basic researchers across India. SIR has also discovered and provisionally patented diagnostic and community screening utility of antigens of malaria excreted into urine of malaria patients, which is being taken up for translation. Along with his team, SIR is developing mass spectrometry-based targeted assays for diagnosis of malaria, dengue, retinopathy of prematurity, and proliferative diabetic retinopathy. Having excellent achievements and a vast knowledge in advanced proteomics and bioinformatics, SIR will enlighten us about the proteomic approaches in medical microbiology. I once again welcome you, sir, and wishes you a greater height of success, and may you bring many more accolades to the nation. Over to you, sir.
So thanks, Dr. Lata, for nice introduction. Thanks, organizing committee, including Dr. Garg, for inviting me for this function. So, can you see my slides? Can you hear me?
Yes, sir.
Okay. So today I'll talk about, I was told to talk about proteomic approaches to medical microbiology. So I will take you through the gist of how proteins and proteomics technology as such can be used to monitor diseases, detect diseases, diagnose and prognose the diseases, with a little bit preference to infectious diseases in today's talk. So I will take you to the genotype-phenotype relationship, which you might have studied in your early studies. So we know a reason behind it. So if we put central dogma into that, so we have gene giving rise to mRNA, giving rise to protein, and proteins, a subset of proteins interacting with each other, maintain a total set of metabolites. That is the biochemistry of that organism or a organ at any given point of time. So the phenotype can also be seen, whether we can consider disease as a phenotype or pathogen which is there in our body. So as a phenotype at that particular time, and metabolites and proteins associated with the pathogen or associated with the disease, which will definitely change the proteome, definitely change for the metabolites. Once you identify what has changed, can we use that for the purpose of investigation on microbiology or medical microbiology? So today the contents of the talks will be brief like this. What are proteins? So when we study proteins, what are the other features of proteins, like structure of protein, function of protein? Then what are post-translational modifications of proteins? These are very important in the context of infection. And how proteins are studied, what are the methods we have to study these changes in the protein. So what is the unbiased way, the hypothesis-free, hypothesis-generating studies like global studies on proteins and its modifications, its features, we call it proteomics. And then we will see how we will differentiate the proteins in a context, in the large context of all proteins with the different features. Then becomes quantitative proteomics. Then what is phosphoproteomics? What is quantitative phosphoproteomics? Can we have a temporal quantitative phosphoproteomics? That means what are the protein modifications in terms of phosphorylation, changes over a period of time after a particular insult is introduced. So then I will go into how these are important by giving you some example studies. So how to study proteins? So proteins are studied mainly by studying the features of a protein. Like any other system, we study the features. We divide its behavior, structure, sequence, and whether there is some conserved patterns in the sequence, and how are they modified, where do we find them often. If I am telling a certain set of protein, whether they are found only in nucleus, whether they are found on the membrane of the cell, or whether they are found in brain, whether they are found in gastrointestinal tissues, so whether they are found in lung or macrophages of the lung, or even the phagosomes of the lung, macrophages of the lung, like that. So what are their association to a particular disease or a particular pathogen? So what are their functions? Do they have a role in disease? Can we study them, use them for the benefit of the society? So what are proteins? Proteins are one of the biological macromolecules, which is made up of amino acids. So linear chain of amino acids is a polypeptide. We can sequence them using mass spectrometry. So these are called building blocks of living system. So we also know about little bit about structure of protein, diversity of the structures. They can be linear then into get into alpha helix and beta pleated sheets, and they are oriented in a three-dimensional structure because of the van der Waals forces and other weak force or strong covalent forces like disulfide bridges, then form a three-dimensional structure called. Then different three-dimensional structure of different polypeptides come together to form a functional unit of a complex or protein. So in the structure wise, there are conserved region in the protein called protein domains and motifs. Domains are a segment of protein that fold into a structure, specific structure, which will have usually have a specific function. Motifs are a short sequence of protein that act as a recognized site for other proteins. So then what are the post-translational modifications? When the protein synthesis is taking place, or when the protein synthesis is completed, other small groups come and add to the different amino acids on the protein. A strong covalent enzymatic modifications happen on the protein, which is called post-translational modification. It can also happen while the synthesis is being happening. So it can be hydroxylation, ubiquitination, lipidation, somylation, phosphorylation. We know around 450 of them today. But unfortunately, the highly studied one is phosphorylation and to a certain extent acetylation and ubiquitination and glycosylation. So mostly because of that, our knowledge on signaling pathways today and drug targets today are largely governed by kinases and largely governed by phosphorylation events, because we had an easy experiment to capture them and protocols were in place. But now you can study many such modification together in a single experiment, which is called multi-PTM analysis, which we are already established several protocols on that. So as far as function of proteins, they can be of diverse functions. You know all of them. There are proteins, when you talk, you think about muscle, you think about hormones, you think about immune system, think about transport molecules, signaling molecules, storage, so many things. They are pleiotropic in their function, although they have a simple 20 amino acid combinations to them. Now, how are we studying proteins? How we have been studying protein? You may remember SDS page, where you just where you can study by running the protein on a SDS page in a single fashion, and then if you have two-dimensional electrophoresis, where you can run it in 2 dimensions so that you can take gel spots and they are separated. So if you add fluorescent dyes to proteins of different kinds and then run and fluoresce them, it can become even a 2-dimensional differential gene expression analysis called 2-DIGE. So the differential fluorescence colors will give you a ratio by the reader, and then tell you how much of the protein has come from one sample versus the other. So these are the gel based techniques, and then we have a gel free technique called mass spectrometry based LC-MS/MS analysis, and also antibody microarray analysis. Now, more recently, the imaging using MALDI has come, mass spectrometry based imaging, called MALDI imaging, where you can differentiate between healthy and tumor tissues and so on. So, if you see in all of them, we use most of them we use an antibody to study the protein, be it Western blot, immunohistochemistry, FACS, fluorescence microscopy, even further. So we use antibody microarray, we use antibody. So there are only two options if you see to study proteins. Either it should be antibody based or it should be mass spectrometry based. To know now the tools to study proteins, the antibody based, you should know the proteins very well to have an antibody against it. Then you also should know the antibody. That is one more level of complexity where you have to know the antibody very well, because otherwise one antibody which works for brain tissue may not work well for lung tissue, or an antibody works well in immunohistochemistry may not work well for a FACS. So these questions can happen. So these questions can we have to be aware of. But then what happens is whatever we study with the antibody-based analysis is always found to be biased, because you know the protein and then you study it. But in case of mass spectrometry, it can give you a global approach where it will not differentiate between known and unknown protein. You will study them, both of them, equally well. So let us come to a basic components of mass spectrometer. What is mass spectrometer made up of? So the main components of mass spectrometer involves an ion source, analyzer, and a detector. Based on this combination of ion source and analyzer, several mass specs are named after them. You can tell EI TOF, MALDI TOF, ESI TOF, or MALDI ion trap. So like that. So various combination of mass specs will become based on the ion source and analyzer. So when it comes to sample processing, so for the mass spectrometry, so this is what I am giving you is for a gel-free approach of 2D LC-MS/MS technique, which we do largely in our lab. So here you have to isolate proteins from any given cell or organism in question, because a lot of your experimental research depends on how you prepare the sample. So you should devise a proper sample processing strategy. So proteins are separated on the gel, and then each gel band is cut into further analysis, which we call as in-gel digestion approach, where you fraction based on the molecular weight of the protein. So molecular weight of these proteins, you fraction them. Same way, so otherwise, in another way, you take the protein extract and then you use trypsin and digest the proteins into peptides, and these peptides are further separated or fractionated into several fractions using various chromatography options. And then, because this is a complex sample, when you isolate a protein, mass spec always picks up things based on the abundance. So abundance. So you have to give less complex mixtures. So to do a less complex mixture, we use this method called fractionation, where you do either chromatography based or a gel based fractionation. Then each sample will be run in the mass spectrometer. Before it goes to mass spectrometer, it will go through another RPLC, HPLC. So very less of all the mass of the same hydrophobic character, same the peptides of one character, all the molecules of that peptide may go into the mass spec at any given point of fraction of a second. And then mass spec measures mass. It separates and measures the mass. And we get a combination of mass of one peptide in a mass spectrometry readout. Let us say that you have given a mixture of peptides. So then at any point of microsecond, one peptide enter into the mass spec. Then it measures the mass of this peptide. So this is very important, this slide. If you understand this slide, you will understand all the phenomena which I am explaining later. So now, this particular mass is called precursor mass. This peptide will be ideally 8 to 12 amino acid because we are using trypsin for cleaving these peptides. Now this peptide, once mass is measured, simultaneously taken into another chamber where it will be collided with the energy which is sufficient to break one peptide bond in this chain. So that results into 2 daughter ions. So now different peptides, randomly this method is done, this peptide bonds are broken. Because of that, you will have all the ions possible combinations from this particular mixture, this particular peptide, and you have that as a daughter ion mass. So you get a molecular mass fingerprinting of all peptides in this way. You have a precursor mass and you have masses of all daughter ions. So it happens in MS level, one level, this happens in MS level. That's why we write always MS/MS in a mass spec. So it is on referred to a peptide precursor mass and all the daughter ion masses. Now once we have a way to represent them from a N terminus and C terminus daughter ions. We call accordingly them B and Y ion, or C and Z ion, or A and X ions. And then usually we get large number of B and Y ion when we use the routine mass spectrometry experiments which we use in our lab. So then you represent them on a mass spectra. So then you calculate the mass differences between the closely situated daughter ions, and that mass difference gives the mass of one of the amino acid. Like this, you will get the sequence of amino acid which is on the based on the mass difference. So finally, a spectra will be decoded like this to have a sequence of amino acids. This is how mass spec works. Once you have a sequence identified, this sequence, several such peptide sequences you will get, and which you will blast against the protein database and say that which of the protein is identified. So now look at the 61 second at 61.2 minute, how the mass spec will look for 1 second. So you take this, a small portion of this, and then you expand, you will have still a lot of masses there. So you will get billions and billions of masses in a single 2-hour run in the mass spec, which you can analyze for lifetime. So usually a biochemist will not decode peptide sequence. We will have a search algorithm which does exactly this mass differences and compares with the database and gives the sequence of peptides, which takes around 20 minutes to analyze one mass spectrum today. So now, using proteomics, using this approach, you can do entire complements of proteins expressed by a cell, tissue, or organ. This we call proteomics. So large-scale study of proteins and their function today in an unbiased way. That means you don't know anything before you do. You know that I want to know that all the proteins expressed in gut, all the proteins expressed in ovary, or all the expressed in platelet or monocytes. So like that. So this particular thing will give you a complement of entire proteome profile of an organism. So we have done for human, which is published in Nature. We have established the first draft map of human proteome in the year 2014. So similarly, now we have told what are the protein complement of a particular organ. What if we want to compare across them? Can we do it in a single experiment? So yes, you can use labeling reagents like DMT or ITRAC. That is, you can label them in vitro. That means you isolate protein, then you do the trypsin digestion, then every peptide, a mass is added. Upon mass spectrometry, this mass is cleaved into 2 and give a signature reporter ion for proteins peptides coming from each sample. So now we can compare these reporter ion intensities in the mass spectra and tell that how much of this peptide has come from sample 1, 2, 3, 4, like this 10 samples in a single experiment. We can compare across 7,000 to 8,000 protein across 10 biological samples we can compare today. So now you can see, so you can have several infection experiments. You can use it for medical microbiology or several conditions resulted from the experiment. You can take any body fluid for that matter, and you can use this on multiple this one, and then you can go ahead with this is called labeling approach, in vivo, in vitro labeling approach. Whereas in vivo labeling, we can use SILAC method, where you can use a particular heavier amino acids, different grades of amino acids, and so that these amino acids which are heavier by 4 Dalton or 10 Dalton are incorporated in different samples, cell line samples, and then mass spec can differentiate between these heavier forms with each other in any sample. So you can do around 3 to 5 different conditions of cell lines. So SILAC approach, you can differentiate them in a culture. This is called metabolic labeling because you are labeling them when the cells are growing, then pooling them together after normalization. You know exactly how much of the amount of total protein you have taken from each one of them. Or you can use cell line based, cell count based approach. Then you can go by comparing across the different types. So now when you enrich phosphorylation, when you do this, when you enrich phosphorylation or any other post-translationally modified peptide from a peptide mixture, we call it as a enrichment protocol followed by mass spectrometry. So that can be very helpful to study proteins which are modified. For example, phosphoproteome, in this entire complement of phosphoproteins expressed by cell, tissue or organism is used. So here you can take away, you will do simultaneously a proteomics experiment, then you do a phosphoproteomic experiment, then we know that whether the phosphorylation, increase in phosphorylation happened because of increase in protein expression or without any effect from the protein expression. Protein expression remains constant, but phosphorylation is increased of that protein. Now, these situations you cannot capture by a next-gen sequencing experiment or transcriptomic experiment. You have to do phosphoproteomic experiment. Most of the kinase-driven phenomenons can be detected only by this method, or you have to do Western blot of phosphoprotein. So now you can identify a large number of proteins which are changing because of a change in a kinase activity. You can downstream signaling molecules which are affected by a kinase or a mutation in kinase can be detected by using this method. So this will be very helpful to understand signaling mechanism because most of the signaling reactions today in any signaling pathways are driven by phosphorylation dynamics. So it is very important to study those dynamics. In a lot of unknown biology will be discovered by using this method. So now, as I told you, proteomics and quantitative proteomics, same thing applies here for quantitative phosphoproteins. Then we can know how much of the proteins are modified by phosphorylation, whether the phosphorylation is higher in infection or a phosphorylation went down during an infection. Because this can be a very, very active therapeutic target if you find such mechanisms in any of this one. Then now let us go one more notch in the complexity. Can we identify at what exact time some proteins are getting modified, phosphorylated or glycosylated or other things? So now you can do it in 2 minutes, 4 minutes, 8 minutes, 16 minutes or 32 minutes. Like this, you can study a signaling pathway in the various phosphorylation experiments. And then you can also see the changed signaling pathways by putting all of them together, these proteins which are modified, you can even identify altered signaling pathway. So variation in phosphorylation at different time points can be assessed by using quantitative phosphoproteomic experiment. So the signaling networks regulated by curcumin treatment, for example, you can tell any other treatment, you can do any other drug for that matter. If you want to study how your drug acts, you can do this kind of analysis. So you can see here, so some of the proteins which are mentioned in red and the other colored things are changing in their phosphorylation level at certain point of time. Which exact site of phosphorylation changes also we can analyze using quantitative phosphoproteomic experiment. We can also then stratify whether they are related to each other by seeing the time factor. So when I put a particular drug, at what time the things have changed, by telling that 2 minutes which molecule has changed, 5 minutes which molecule has changed, 32 minutes which molecule has changed, you can arrange them in a order. So then coming to another method using mass spectrometry, this is a method to validation or a targeted proteomic analysis, just like Western blot or just like quantitative PCR. You can study the variation in the protein amount using a known protein, but you don't need an antibody for that. So you can use, there are 3 different methods called multiple reaction monitoring, data independent acquisition based SWAT methods, and also a parallel reaction monitoring. All these methods are used to study a known signaling molecules or a known molecule and how much is the expression, how much is the modification. We can design experiments, we can come up with a panel. The advantage of this is you don't need an antibody for this, and you can also make them as a panel so that they can be a diagnostic assay for future. So now when it comes to proteomics based biomarker investigation, you can study tissue, you can study secretome of a cell line or a organ or a infected cyst or anything. Then you can also do proteomics of biological fluids like sputum, serum, CSF, urine, saliva, tear, bile. We have established a method for all these kind of body fluids today. We can identify proteome, and you can even detect proteins of infectious etiological agents using all these body fluids. You can also do phosphoproteomics, you can also do glycoproteomics, you can do multiple post-translational modifications. So now let us come to specific area of medical microbiology or infection biology. So let us say chronic meningitis. See, all chronic meningitis look alike. So, for example, tuberculous meningitis, cryptococcal meningitis, toxoplasma meningitis. Their mechanism of action is different. So, the prognosis is different, and drugs of choice are different. Treatment options are different. But you have to detect them early. And you can also study what damages they cause, what different molecular damages they cause. You can also come up with a new way of treating them. So, for example, tuberculosis and sarcoidosis. An early discovery will help. Both look alike. One is autoimmune disease, another is infectious disease. Paralytic rabies and GB syndrome, another perfect example of how things will look like. GB syndrome patient is suspected to be paralytic rabies and put it in ICU, and we don't have a definitive marker today to distinguish them. And then you can also have antigens of pathogens present in the body fluid. With this, you can do a lot of diagnostic cases, detect the pathogen. And importantly, one amino acid change also can be detected in mass spectrometry, which can even tell you the mutation status of these particular, drug resistance status of this particular, or virulence status and persistence status of these pathogens, if you already know the biology from genomic studies. So you can also do proteogenomic study of microorganisms, where you can annotate the genome of various microorganisms using proteomic data. So we have done it for a large number of model microorganisms, from Mycobacterium to Cryptococcus to Candida, and even Leishmaniasis and etiological agents of Leishmaniasis. So like that, and so on. So many microorganisms we have worked on. We identified 40 to 100 to 200 new proteins which have not been identified so far by genomic studies have been identified using proteogenomic study. Today I am not going to dwell too much amount proteogenomics. You can go through our website and you can find out. Then you can also study phosphoproteomics of virulent and avirulent isolates to study whether there is any phosphorylation events or kinases are involved in the virulent process. And then if there is, that can be an active drug target to stop the virulence. And then you can also do targeted proteomics of pathogens itself can come up with the MS-based diagnostics. For example, here, so tuberculous meningitis, which are the molecules in the brains which are changing when the tuberculosis, the Mycobacterium tuberculosis infects brain. So that we can identify like this by a quantitative proteomic experiment. There are here several proteins and you can see the ratio of labels. These are chemical label reports I have told you. Then you can validate many of them using conventional immunohistochemistry. You can also compare across different etiological agents, how they are changing the protein expression pattern, and is there a particular proteins or signaling pathways are affected by different etiological agents. Of course, there are. So then you can also identify whether there is some markers available in a body fluid for which tells that, oh, this body fluid belongs to a TBM patient or this body fluid belongs to a pyogenic meningitis patient, something like that. We have identified several of them. And then we also identified several markers, blood-based biomarkers for GB storm and paralytic rabies. There are a large number of proteins are coming up. Each protein can be used for identifying a diagnostic assay or coming up with a diagnostic assay. And then now rabies. Rabies as such, the virus doesn't kill cell lines. So brain also remains intact. Then how the rabies kills? So when we did gross brain analysis, gave very few proteins which are changed by rabies infection. These are involved in endocytic recycling and synaptic vesicle exocytosis. Now these are the drug targets now for the rabies virus. Something is happening in this area. A small set of molecules are actually causing problems in rabies infection. These kind of things leads you will get if you do have proteomics based experiment. Now, what are the changes happening in a chikungunya patient? So let us say there are proteins, very few proteins from this analysis you can see is changing in the proteomic experiment. Large majority of the proteins remain same. Only few proteins are changing, and these extreme proteins which are changing can be a biomarker or can be used to study molecular mechanism. So when we studied this in a serum of chikungunya patient, so we are seeing that the fatty acid metabolism and calcium metabolism of apolipoproteins and S100 proteins are highly affected by altered in the network of protein altered in chikungunya versus other diseases. Like you can compare chikungunya with dengue, malaria, then you can come up with proteins, why certain proteins are expressing more or less like that. Then you can also do phosphoproteomics of avirulent and virulent strain and identify proteins which are involved in virulence. We have done with the F37RA and RV. We identified large number of proteins which are changing, and very interestingly, these are all in the area of the secretory system of microorganism. So these are the very new knowledge we added to the microbiology of tuberculosis. So which is the virulence factors, how they are changing in their phosphorylation pattern by doing a quantitative phosphoprotein experiment. Then doing a proteogenomics and phosphoproteomics and proteomics experiment, we also identified molecules which are actually changing virulence in F37RV, because large number of proteins which we are calling as virulent genes which are absent in F37RV actually present in F37RV, because long back when we did genomics, because of limitations in the genomics, we lost these 12 or 13 genes, but they are expressed from H37RA, but only a small set of proteins today and the phosphorylation pattern actually gives virulence to H37RV when compared to H37RA. So we redefine the virulence attributes We change the medical textbooks when we do these kind of experiments. So these are all published. We also did Plasmodium falciparum when it is burst out every 48 hours, and the merozoite um, um, going back to the red blood cells, enter erythrocytes. So what are the proteins which are actually mediating signaling during that stage? So can we calcium dependent protein kinases in Plasmodium can be a target for clearing merozoites from the blood, and so that malaria is clear. So we also did a whole genome sequencing of MTB. I almost covered the time. Now I will just with the 2 minutes I will close this. So we have identified a lot of known drug resistance markers in Indian population, but in addition to that, we have identified several other mutations which are specific to Indian varieties, Indian clinical isolates, which were not known in the public until then. Then we also identified several mixed infections, which were supposed to be Mycobacterium tuberculosis, but there are so many other Mycobacterium NTMs in the mixture. So by seeing this, we have identified on 343 novel SNVs in the 38 known drug resistance genes, which we published. And we have even patented their utility for detecting drug resistance genes in India. So now we are developing a DAA based assay for, because even one mutation can be recognized in mass spec variant peptides and putting normal peptides in the database. So we are detecting these things and we are developing an assay right now for detecting drug resistance MTBs and NTMs. We are also developing a protocol for detecting dengue viral proteins of different strains in any mixture, and then we will keep adding other acute febrile illness pathogen antigens into this assay so that it will become pan acute febrile illness assay identified through mass spectrometer. We can also identify urine, using urine we can identify whether it is a Plasmodium falciparum or Plasmodium vivax or a mixed infection. We have peptides which are excreted to urine. We identified them in the preliminary assay. Now we are developing a diagnostic assay for this, which is now funded by DHR and approved by DHR. Money is yet to receive. So we have even patented these peptides for diagnostic assay. And we developed a multiple reaction monitoring assay for already we have developed and using mass spectrometry. And then there are a lot of interdisciplinary area can be worked out. Once you generate this kind of data, you can do signaling pathways work, you can do proteogenomics, proteometabolomics. It's a new area, and then proteomics diagnostics can be used for well, this. With this, I thank from where I left Institute of Bioinformatics. We were a very small group of scientists there, worked on various areas. Then we established a proteomics and bioinformatics facility in NIMHANS at NIMHANS IOB lab, where we procured a arbitral fusion trade mass spectrometer. Recently, we established these facilities in Yanapoya University in Mangalore. And then I thank all my collaborators, mentors, and team students. I acknowledge their contribution in all the things which I presented. So various institutes have collaborated for making it happen, including Goa University, where I work for malaria proteomics in collaboration with National Institute of Malaria Research. And I also thank various funding agencies for funding these efforts. Thank you one and all.
Thank you very much, sir, for a very informative talk and enlightening us about the significance of proteomics, particularly multi-omics approaches in monitoring, diagnosis, and detection of various infectious diseases and
Thank you.
neurological diseases. Now, I would like to ask Anybody? Uh,
Hello, Dr. Keshav
yes, sir.
Prasad.
Hello.
Nandakumar, sir. You can
Hello,
go
sir.
ahead.
Nice
How
to
are
see
you?
you after decades. I think I am very pleased to see you and congratulations and welcome to this webinar.
Thank you. Thank you.
I'm happy
Nice
to
to
see
see you
you
after a long time.
face to face. I think it will not be possible because you know that there is a voluminous research that you have been doing for last, I think, at least 20 years. And I have pleasant memories of association with you in Goa University.
Thank you.
And you are always in our heart and
Thank
mind.
you. Thank you so
My
much.
simple questions are, you know, that mostly in applied part of the research because the structural and functional proteomics is a advancing area now, and with new tools and also the tools that you are developing, definitely it will benefit the human welfare. My specific question is, you see that what are the leads that have helped, and you will be providing leads to various, I think, applications like you see epitopes for design of vaccines or immunodiagnostics. So could you give some examples, Dr. Keshav Prasad, of the outcome of this published research and the leads that have been generated through your proteomic analysis for immunodiagnostic, that applications of diseases?
So I can already tell you that one of the phosphoproteomics experiment we have done on curcumin led to the identification of CAMKK2 as a lead target of curcumin. Now we are taking it to clinical trial. So CAMKK2 inhibitor can be used effectively for as a therapeutic target in gastric cancer. So this has directly happened because we are the one first identified CAMKK2 as a lead target of curcumin and can be used in treating cancer. So this has already developed into a therapeutic approach. Then we also identified another molecule now in by using a traditional drug as a surrogate. We identified a molecule in treating Parkinson's disease. So that also now is being developed in clinical trial for the further development. And we have also identified the target of Annona muricata, Lakshmanapala, which people use as anti-cancer drug. And that is also now somebody can take it up for the trial. And then as far as you are telling immunodiagnostics is concerned, now we have identified peptides and we have put them in the public domain. We have no idea who is developing what based on these results. These are all in public domain. So once they develop, they patent it, the utility of that. But we have not gone and actually documented any one of them, that whether that has come because of our study or somebody else's study. So, but we are developing, as I have shown you, a urine-based diagnostic test, immunodiagnostic test, to be precise, just like a urine pregnancy test we are developing for malaria now. This is particularly useful not only for malaria patient, this also be used when you wanted to do malaria eradication program. You also have to identify a non-symptomatic people in a community screening. You should not have even non-symptomatic malaria people around you if you want to really identify malaria diagnostics in the malaria eradication program. In that, it will be highly useful, this tool, because it doesn't need blood, it needs a drop of urine, which will tell you whether that person has harboring malaria pathogens or not. So now going forward, the same immunodiagnostics, we are now improving upon that by adding dengue antigens into it, chikungunya antigens into it, and other JE and other acute febrile illness antigens like leishmania and leptospira into that, so that we can come to know in the beginning itself whether the patient is suffering from leptospirosis, because treatment is simple. Treatment is very simple for leptospira and leishmania if they are, scrub typhus if they are detected early. But once you allow them to go detection later stage, they are already create multi-organ failures. Then you cannot treat it. So that is why we have to identify a person when he has fever. When he walks in, immediately we should identify whether they have streptypus or leishmaniasis, streptypus or leptospirosis, as opposed to dengue, malaria, and other diseases. So these are being done now in our own lab. We are now having a big translational program on these things.
Hello, supplementary question. You see that this question is little tangential, and I know that you could say that this is not the time to discuss it, and the answer is not in my hand, and all those things. But it's like this: that you have a critical mass of now knowledge and that infrastructure, right, and technology, where you are working, and you have collaborations also which are supportive and complementary. The question is, you see that there are now new challenges coming up in the area of structural and functional viral proteomics in view of the present pandemic and the pandemics which are likely to follow. We do not know exactly when. So I know biosafety considerations and the requirements for BS level 3, 4 labs, and the maintenance of viruses, and all these concerns are there. It is understood, but and is there any plan means to combat those challenges and accept those challenges, that using the critical mass of knowledge and technology that you people have developed, your team has developed so well, and collaborations also, that is there any plan to jump into or join this area of structural and functional viral proteome research? I am talking about human, those viruses which infect human, means zoonosis, which is caused by the viruses which humans. So is there any plan, or you have restricted yourself to non-virological areas specifically because of your priorities of research and funding? That's all.
It's a very, very pertinent question at this juncture of the crossroad where we are staying. Thank you for raising it. See, I have no caste-based or let us say a group-based priorities. Maybe it is virology, botany, microbiology, veterinary. I have nothing like that. See, I am a technologist. I have gained this knowledge by using my time. I invested a lot of time. If anybody benefits, any collaborators benefits on that, I am ready to pitch in. But particularly those which are required BSL-3 and BSL-4 setup, it is highly difficult because we don't have such a huge number of facilities which can cater to the need of research. So in India, because most of our BSL-3 and BSL-4 facilities are dedicated for public health priorities. So there are hardly any time. So once they are dedicated to public health, it becomes very difficult for researchers to get into the room and do his research, because already the standardization GLP protocols prohibits large-scale research inside such laboratories. So in India, we have to come up with large-scale, programs for research in this area. One particular thing in the context of TB, because that also required a same kind of setup, PSL3, and it is a very slow-growing organism. We get very, very low quantity of protein to start with. So we have developed this DIA-based proteomics. So DIA-based proteomics is nothing new. In fact, when the mass spectrometry developed, it was DIA-based. Later, DDA has come, data-dependent acquisition has come. Now we are forgotten how the DIA has been done. So now they have come up with a new name called SWAT and all those kind of things for these analysis protocols. So just to sell them. So now we can use such facilities and we can do with the very, very less protein availability, less mass of biological material, we can do a lot of work. So that I am developing now. I am looking for virologists to help me. I have applied a lot of projects on COVID also during COVID pandemic, and I have even thought of establishing a BSL-3 facility in Yanapoya itself. It's a work in progress, but it is very difficult to get into that because of public health priorities of most of the virologists and other people. Research takes a back seat. Researcher keeps on waiting for samples as well as funding. It never happens.
Any other questions from the participants? Yeah, I think no more questions. Thank you very much, sir, for sharing your such a vast experience and knowledge about this approaches or the applications of various techniques like proteomics or genomics for monitoring, diagnosis, and detection of various infectious diseases and neurological diseases, and also for explaining in simpler language about the basic functions of proteins and explaining about the high-end and specific sensitive technique like mass spectrometry and its applications in disease detection. So thank you very much for sharing your time, especially, and enlightening us with your knowledge, and for a fruitful discussion with your knowledge. Thank you very much, sir.
So thank you very much for organizers for inviting me, Dr. Sandeep Garg and Judith and Dr. Lata for taking this session, and Dr. Kamath, Nandakumar Kamath, after a long time. Nice interaction. Thank you. Thank you all people who patiently hear my talk. Thank you.
Thank you, sir.
We will now take a break for lunch and join again at 2 o'clock.